Gavin is a clinical specialist and head of the cancer genomics team in the Newcastle genetics lab, based at the International Centre for Life site. His team deliver cancer genomics testing to the team of haematologists in the northeast region, and Newcastle was one of the first centres in the UK to replace FISH testing in CLL with SNP array testing, and that's not political. He's also going to try to explain the difference. Now, I've seen this presentation. Buckle up, folks. This is...
John, thank you very much. Helen, thank you for recruiting me. It's a difficult thing to explain what goes on in the lab. So our lab is just down the road at the Centre for Life. You know, there are other things other than the ice rink there, so there's a team of NHS staff there. And we spend so much of our time talking about genetics. We're all genetics geeks. It's a really sobering thing to try and make this presentable, right? So I'm probably going to fail at that because it's so complicated. But let's give it a go. Helps enormously that you've talked about IGHV, yeah. So genetics. I mean, we're in an amazing room here. It seems appropriate for me to put Da Vinci's Vitruvian Man up, yeah, as a representation of a person. So. In the cells of a person, every cell has got the entire set of genes that make up that person. The more you look into this, it's astonishing. So each cell has 46 chromosomes. Each of those 46 chromosomes, well, the 46 chromosomes together have got 20,000 genes, and they code for proteins that build stuff and do stuff. So I'll keep using an analogy all the way through this, yeah, that a gene is like a book. A gene is a book of instructions that then you go off and make things. It's the instructions to make are like a Lego thing that then goes off and does things in the body. Chromosomes are like a whole load of those genes all together, yeah? So I mean, we're close to the Littenfield, yeah? If you've not been to the Littenfield, you've got to pop your head in. It's an amazing place. But let's imagine we're in that library there. Each book is a gene. Each chromosome is an aisle within that library, and the library's got 46 aisles, yeah? So that's sort of like where we're going. And I just want to really clarify that there's two sorts of genetics. Helen, you were alluding to it. You naturally think of genetics and you think that this is something that I'm going to pass on to my children. It's something that you're going to inherit, and you'll be familiar with a lot of genetic syndromes and disorders that we deal with at the Centre for Life, like Down syndrome, cystic fibrosis. All of these are what happens if you've got a genetic defect in every cell of your body. In there, we call those constitutional abnormalities or inherited disorders. Every cell of your body, generally speaking, will have that same thing. So in Down syndrome, that's an extra copy of a whole chromosome, yeah, a whole extra copy of chromosome 21. In cystic fibrosis, it's just a tiny little mutation. So it's an enormous scale that we operate on, and the key thing there is that with those inherited disorders, you can pass those on. Those abnormalities are in sperm cells, they're in egg cells, so you can pass them on to the next generation. Forget all of that, yeah, because that's not what we do. It's the same thing. We're talking about books, we're talking about aisles, we're talking about genes and proteins, yeah, but we are just focused on, our team, our cancer team, are just focused on using genetics to get really useful information for our clinical colleagues to identify which sorts of CLL are going to respond better to treatment, which sorts of acute leukemia, let's say, we need to treat with different things. So these abnormalities, so I'm talking about P53, I'm talking about IGHV, and I'm talking about other deletions and things. They are just in your blood cells. They are just in your lymphocytes. They're not in your egg and sperm. You can't pass them on. And it's really easy to get confused between the two then, yeah? So I think Helen has beautifully started us off on that one, actually. So I'm going to try and talk through some of the tests that we use. So one test that we use is called FISH. You might have heard of FISH. It may be some surprise to you. There's no fish involved. It's not anything to do with fish. It's just an acronym that stands for fluorescence in situ hybridization. It just means putting coloured probes on cells and seeing how many of them there are. It's basically that. Chromosome analysis. You know, I said we're all genetics geeks, right? I'm specifically a chromosome geek myself, yeah? So we just love chromosomes, some of us. Now, we'll talk a little bit about how that works. It's just useful to see all the DNA. When I show you chromosomes, it's like you're looking at the whole lot. I'll come on to that in a second. SNP array is a really detailed way of looking at chromosomes. I'll try and explain that, but most people in our department can't explain SNP array, yeah? So we're not going to go into very much detail in that, but it works brilliantly. And then, sorry, I'll keep showing that sort of diagram on the right then, yeah? Because I just think it's really useful to always, we're operating across these different scales. There's the chromosomes, the aisles of the library, and then there's the base pairs, the genes, the books of the library. So within each book, each gene is a book, yeah, and it's got loads and loads of pages, and the letters on the pages, they're the A's and the T's and the G's. They're like the base pairs. The genetic system reads those, and that becomes the sort of Lego model that does things in your body. Okay, so I'll just try and give you an idea of how that scale works. Now, we've talked about TP53. I'll focus on TP53 because if you're looking at all the chromosomes, it's over here, by the way. It's on chromosome 17, and yeah, it's one of the things that a lot of us in the lab do. We still do this manually. We look down a microscope and try and identify, oh, sorry, I've got like a little automatic thing there. Let's just imagine that didn't happen. So normal chromosomes, you've got one from every parent, one from each parent, yeah? So you have 23 from your mum, 23 from your dad, and that makes the 46. And we look at them manually down the microscope. It's going to do like a little sort of like a wiggly fade in. So that's what you might see if you looked at lymphocytes from a CLL patient. So can you see there that we've just chopped a little bit off the end of chromosome 17? That's a 17 P deletion, we would call that. Chromosome 17 has got two bits, a short arm and a long arm, yeah? And there's a little bit been chopped off the short arm. So that's exactly the sort of thing that you might see if you'd got a P53 deletion. So let's just talk about the P53 gene for a second. So the P53 gene is on the short arm of 17. I'll talk a little bit about the function, but it's critical in identifying DNA damage, and its job is to identify DNA damage, collaborate with some of its other protein friends, and shut down a cell that's got DNA damage. Generally speaking, cells replicate, and you want the cells, the daughter cells that are sort of like created from it splitting, to be exactly the same. Everything needs to be exactly the same, because if you start accumulating mutations, then that's the way that could develop into cancer. So chromosome 17, that's like an aisle of the lit and fill, yeah? The P53 gene, which is there on this diagram, is the book. And let's just imagine for, I just want you to go with me for a second on this one. Let's imagine the P53 is that big, yeah? P53 is 1 meter long. And then if that is the case, it's got 25,000 base pairs, yeah, within that 1 meter. So on that scale, each of those base pairs, the A's and the G's and the T's, is like one hair, like a hair's breadth along a 1 meter length. So so we're here, we're in the common room, we're in Newcastle. That's like an aerial photo of Newcastle. I'm just going to use this just to give you a sense of scale. If P53 is that big, yeah, then on that sense of scale, with us being centered, that's where we are. The short arm of chromosome 17, which is often deleted in CLL, that would stretch all the way to St James's Park then, yeah? So the amount of scale between base pairs, which is like a hair, and the whole of the short arm is enormous. So chromosome people, we tend to focus on like the big scale, but we cover the whole range. If you look at the whole chromosome, that would go to the metro centre. If you stuck all the chromosomes together, yeah, they would stretch to Leeds. They would nearly stretch to Edinburgh. To be honest, John, I've got Aberdeen on here, and I've only used one of each chromosome, so there's two of each chromosome, so they would actually stretch to Aberdeen. So that's incredible. That's one gene, and then the number of those and the scale is enormous. And it's because the DNA is so compacted. It's wound and wound, and then round again in a super they call it supercoiling, that makes them fit together on these chromosomes. Okay, so just coming back to P53 then, that's P53. It's 1 meter long and it's a book. It's on chromosome 17, which is an aisle in the library, and then it makes a protein. That's my little symbol there to indicate that it makes a protein. So that's just an instruction book to make a protein. The protein is so important because it goes around the cell, it looks at DNA. If it identifies that there's any damage, it binds to it, and then it communicates with other proteins, and the cell gets shut down. It's a way of stopping DNA damage. So it's a way of suppressing tumours, because a tumour might develop if you've got accumulation of DNA damage. So the TP53 gene is a tumour suppressor gene. So if you don't have that, you can't bind to DNA. So the thing on the right is that's P53 wound around a bit of DNA. It can't do that. So broken DNA might accumulate, chromosome abnormalities might accumulate, and that might lead to tumour progression. I'll just quickly whiz over this because I know I'm going to be short of time, but it's interesting to think that there are two P53 genes because you've got two chromosome 17s, yeah? Remember, you've got one from your mum and one from your dad. So you've got two aisles, and both of those aisles have got a P53 book. So when you look into the detail of it, actually, you need to get rid of both of those in order for the DNA damage thing to be a problem. Because if somebody steals one book from the library, yeah, if you delete one copy of P53 from that aisle, you still got the other one next door, the one from your mum or your dad. So that's tumour suppressor genes. They come with a natural understanding that you have to take out both of them. So sometimes it might be a deletion. So a deletion would be if you go to the library and the book's not there. Somebody's stolen it, somebody's not returned it. There's no P53 book, but it could be a mutation. Somebody could have scrawled all the way through it, so the instruction doesn't make sense. And so when you try and make the Lego model of the P53 protein, it's all wrong and it can't bind. So deletions and mutations are just different ways of not having P53. But we look at them in different ways. That's back to the chromosomes again. And I'm just talking about really big things, yeah? So that's a big deletion. It's taken out half of between here and St. James's Park, yeah? And we use fish to look at that. Well, we used to, we don't do it anymore actually, but a lot of labs still use fish. And so you effectively label the P53 gene red. So on normal 17s, you've got two P53s, so you'll have two red dots in a nucleus there, and what you're looking for is a loss of one red dot. You're looking for one red and two greens. So that's how you do P53 fish, and that indicates that P53 has been lost. In most cases, you assume that there's been a mutation on the other one as well. So this is what it looks like down the microscope. This is, people in the lab love doing fish because it's you have to turn the lights off. It's all like atmospheric in the lab. We're looking at nice colours, yeah. A lot of the people in our lab like looking at stuff. So it's really visual. So that's a normal cell. Don't know if you can see my mouse moving, but that's a normal cell because you've got two reds and two greens, yeah. That's a normal cell. Two reds and two greens. I've just highlighted which ones are not normal. All those with little rings round have got one red and two greens. That is what you're looking for if you've got a P53 deletion. Now, the fish is great. It really works, and it's really quick, and it's a nice cheap test. So, but let's imagine if that's the P53 gene. Yeah, it's 1 meter long. The fish probe is actually, it's huge because you need like a load of the fish probe in order to be able to see what you're looking at. So actually the fish probe will be the width of this room, yeah? So you could have like a little bit chopped out of your 1 meter P53 gene. You could have some pages ripped out. You could have a few pages ripped out, and the fish probe will never see it because the fish probe is just quite happily spanning this room. So we wanted a more sensitive test. So let's imagine you take all the chromosomes, yeah, and you chop them all up, take the whole thing and you put it in a blender, yeah? It's a bit like that actually. So you fragment the DNA into little tiny bits, And then we've got a technique called SNP array. It stands for single nucleotide polymorphism. I'm not going to spend long talking about this, but instead of, and some of those bits of DNA have got little labels on them, or we call them SNP features. So going back to the P53 gene, that's the P53, that thing there on the screen, that is my 1 meter P53 gene. Instead of the fish probe spanning this room, that 1 meter has got 11 little dots on it, yeah? So we can actually see smaller abnormalities by whether or not those dots have gone. So if somebody had just ripped out a few pages, or they'd ripped out a chapter, then we're able to see that with SNP array. The beauty of it as well is it looks at other chromosomes as well. I'm not going to spend long on this because this is just showing you what this test looks like. It's less visual, but it's still quite visual. So whenever you see the way our technique presents itself, if you're just seeing a flat line, that's normal, yeah. So instead of the chromosomes looking like chromosomes with black and white stripes, remember, we've chopped it all up and we're just counting dots now. We're just counting dots. So look at that there, when you've chopped up a 1 and then you just count all the dots. A line in the middle means you've got 2 of everything, yeah? 2 of everything is right, 1 from mum, 1 from dad, 2 of everything. So then look, so that instantly that chromosome there, that's chromosome 10, that's all wrong. So there's a lot of chromosome 10 been deleted. Chromosome 13, the most common thing that happens in CLL, the majority of patients with CLL will have 13q deletion, and they're often really small like that. So that little dip down there means that a little bit of chromosome 13 has been deleted, and SNP array allows us to really easily see that. So that's chromosome 17. We're back to 17, about P53 again. That's what 17 looks like on chromosomes, but then that's what it looks like on SNP array, yeah? This bit, all of this bit, there's 2 copies, and then there's the short arm is just gone. It's dropped. It's the fact that it's just such a really clear, elegant way of demonstrating it. So that's the way that we now look for deletions and other copy number changes in CLL, because we can see a 13 deletion, the 17p deletion, a P53 deletion, trisomy 12, 13q deletion, 11q deletion, all in one test. And then, so this is an unnecessarily complicated slide, but you remember I was talking about that there are 2 copies of chromosome 17, so there are 2 copies of P53. So if you take one out, so somebody's nicked that book, there's still the other one in the other aisle. So generally speaking, you have to, in order for P53 to mean something in your cancer, you basically need to have something affecting both of them. So maybe both books are nicked, or one of them has the pages ripped out and the other one's gone. So there are multiple different ways that you can look for those different abnormalities, and one of the most significant is by actually sequencing them, yeah? Getting that book, looking at all of the base pairs, so reading the book, the A's and the T's and the G's and the C's. So that's the P53 gene, and if you do that, there are some areas of it which often are just, things go wrong in those areas. So and we call those mutations, yeah? If you read a gene and there's something wrong with the text of it, that's a mutation. So now we're down here. We're solidly just looking at the DNA sequence, and so that can lead to, you might make a P53 protein, but it's the wrong shape, yeah? So it can't bind to DNA, or it might be missing bits. It can bind to DNA, but it doesn't communicate with its friends. There's a whole load of reasons why a mutation might affect the function. So that's an awful lot of what we do in the lab now, is looking at a mutation and what's the effect of that going to be on the protein. Okay, so we collaborate with our colleagues in Leeds. We work together with Leeds and Sheffield and the Newcastle lab, and in this situation it's our colleagues in Leeds that do the sequencing for us. So we do the thing, we take samples, we extract DNA, so it's basically putting the chromosomes in a blender. Then every day we've got a van that takes stuff from Newcastle to Leeds. Leeds put it on a fancy sequencer. There's an awful lot of fancy machinery and genetics that's trying to do this, and that generates that base pair sequence. It reads the book and tells you exactly whether the instruction manuals are right, and we look for P53 as well as other genes. Okay, so I'm hoping I've had a reasonable go at covering fish chromosomes and mutation testing, direct sequencing. I'll just go over IGHV again, just in an attempt to just about be on time. So IGHV, again, this is done for us in our region. This is done by our colleagues in Leeds, and this is an unnecessarily complicated version of the so the sort of pathway that Helen showed is it's the representation of a B cell going from a stem cell within the bone marrow where cells are made before they go out into the blood, and then moving all the way forward to like a fully differentiated B cell that's producing antibody. That's the critical bit of the immune system. It produces antibody that fights infection. So as Helen described there, as a critical moment in the lymph nodes where somatic hypermutation happens. So all cells from this bit, from the first bit, stem cell, maturing cell, IGHV is unmutated, and then that once they've been through the germinal center reaction, you've had somatic hypermutation, they're now mutated. It's a weird thing to think about, but that's completely normal. You know, you think about mutation and something's gone wrong. No, this hasn't gone wrong. It's fundamentally part of what. So messing about with that book, the instructions for IgH, is completely normal, and it's the way that we fight off infections. It's the way that we've evolved to do that. So that's the immunoglobulin molecule, and the IgH bit is the bit in the middle. There's bits on the side. We're just looking at the bit in the middle. It's put together by different bits of Lego. And the hypermutation just makes changes in the fingers of that. It's like two hands, isn't it? And then the fingers of that. It makes loads of different variant forms of the fingers of that. And that, so the way that Helen described it, of B cells being taught how to react to antigens, to invading foreign agents in the blood and the body, is because you've made loads of different sorts of fingers, and then the right fingers attach. Some get thrown out, some get selected. It's normal. It's normal to be mutated at that particular point in development. So when we talk about IgH, it's a different chromosome. It's only me that's interested in the chromosomes. That's chromosome 14, and IgH is right down the end of that then. And then so we sequence that. Our colleagues in Leeds sequence that. We send samples, and our colleagues in Leeds sequence that. So this is, I'm not expecting this to make any sense. It's a little bit of sequence. The green bit is the critical bit of the IgH fingers. It's looking at those fingers and seeing whether or not there's been any changes. And then you compare that to a reference sequence. We do a lot of this in genetics. There's an awful lot of databases and comparing with things. Is this the right sequence? So I don't know how clearly you can see that, but there's 16 yellow little crosses on there. So in that little stretch of 200 odd base pairs, 16 of them are wrong. That's and we because mutation is such a rare thing. That's a lot of mutations in like a short bit of DNA. So that's what we call hypermutated. That would be consistent with us saying it's a patient with CLL where their clone shows IgHV mutation. You only need to be below 98% in order to say that, actually, because mutation is just in other settings is super rare. And the other weird thing is that that's normal, and it's with traditional therapies is associated with a better outcome. It sort of reflects the fact that the further away a cell gets from a stem cell, they become almost closer to naturally dying. Stem cells, you could imagine as sort of immortal. I mean, I'm not a hematologist, Helen, so apologies. We try and come up with these things to make life simple for us. Let's imagine stem cells are immortal, and then when cells actually are fully differentiated, they have a very short lifespan. So as you go along that pathway, the cells become easier to treat. If it's a leukemia that's derived from them, they become easier to treat, because stem cell leukemias would be very hard to treat. So finding an IGHV mutation marks you as being down here. You're down this end, you're more differentiated, the cell is, it's easier to treat on the old therapies. And then unmutated, you sequence it, there's nothing wrong, 100%. That's the way our result is presented, 100%. We'd report that to Helen. Unmutated immunoglobulin, so you're down here. this is the cell that's originated, the CLL clone in a patient like this has not been through the germinal centre. It's closer to a stem cell on the traditional therapies that might have made that harder to treat. Right, thank you very much.
I'm glad that's cleared that up then.
What I'm a bit confused about is that we've heard that it's not something that's passed down from your parents, so it's not something that you inherit, but then we're talking about genetics and DNA and mutations. So how does that, why do we get it? If it doesn't come from our parents, why do our genes turn in on us, if that makes sense?
Yeah, because we use the word genetics to just describe the normal process of just cell growth and development.
Right.
Because all cells in our body are dividing all the time, and that process is genetics. Yeah, that is genetics. When your hair grows or as you grow up, it's because your body is replicating its cells and making more of them. So it's just the natural way that the body grows and cells divide. And so if something goes wrong in that process, nothing to do with passing anything on to your children, if something goes wrong in those abnormalities, a mutation that you might pick up could make a cell maybe more active in division or less responsive to controls, or various other reasons why a cell might become more like a leukemic cell. Yeah, but it's very often genetics that matters. It's something about that cell dividing. So even though genetics, we've got it in our head, haven't we, that genetics is about inheriting things, genetics is what's going on all the time, because our cells live and develop by duplicating themselves, and sometimes, very rarely, it goes wrong. And then you'll make something will go wrong. You'll make two cells that are dividing a little bit faster, and then they all start to divide a little bit faster. That was a genetic abnormality because there was cell division happening there, but it's not affecting any of you.
So the chromosome 17 deletion, do we know why? Just don't know why it happens then.
No, we don't know why. No, that is that
would be brilliant.
That is the complete mystery behind everything, is how these happen. So DNA damage happens, cosmic rays, X-rays, radiation, things like that. Some things like that will affect DNA and cause damage, and then P53 and other genes like that are sort of like identifying cells and shutting those ones down. Sometimes something gets through, and P53 is maybe not active, and then you get a mutation, and then a few little steps all combine, and then that leads to the development of a leukemia.
12 years after a bone marrow transplant, a relapse. Simplistically, when Paul had his, you just imagine that he was completely wiped out of all of his cells when he had his first lot of wipe out the chemotherapy. So does that mean over that time, Paul's his own DNA that I thought would have been completely wiped out has somehow managed to get back in and cause the relapse? Is that how it works?
No, it's a good question. And one of the things that we sometimes, definitely after a bone marrow transplant, we measure this thing called chimerism. So first thing to say is bone marrow transplants in CLL these days are incredibly rare, simply because, as we've said, in terms of the treatments we've got nowadays, we often don't need stem cell transplants because we've got treatments that can keep people in remission for many years, and then we've got another treatment we can use next, and so on. But because we have used stem cell transplants in the past, and for certain patients, for example, Liam, we will still use transplants going forward. So if you've got a stem cell transplant, and quite literally you're getting cells from somebody else, and then those cells from somebody else, because they're their stem cells, and as we said in our hemopoiesis bone marrow thing, so we're getting somebody else's new, completely immature stem cells to then go and hang out in our bone marrow and make all of our new mature blood cells from there going forward. And so what you're hoping for there is that all of your blood cells in your bone marrow will all be taken over by the new donor cells. So it's making your immune system and all of your normal blood cells. We know that sometimes some of the original blood cells can hang out at a very low level. And so it's like there is a balance there between needing those new donor cells to be the ones that are now in charge and old cells not even there anymore. But there's the other side of that, as you would have known at the time of a bone marrow transplant. We talk about this graft versus host disease, because we've got a new immune system coming in that then recognises all of the patient as being foreign and starts attacking the whole patient. So we have to balance giving something to calm down the new donor's immune system so it doesn't attack the whole patient, while still letting that immune system be active enough that it takes over all of the blood making and wipes out the old blood making. That balance can sometimes, just the difficulty of achieving that balance, sometimes we can have just a teensy little bit of residual something that can then start to grow back in the future.
Sorry, loss of ATM. What's that signify?
So I mentioned a couple of times about 11q deletion. ATM is another gene that's associated historically with CLL. It's harder to treat, and that's on chromosome 11. Yeah. So another chromosome, another gene that is is associated with poorer outcome.
Just to clarify something, your mutated status can't change, but can your deletion change from a 13q to a 17p?
Yeah, as Helen referred, the CLL is a clonal disorder. So you grow from one cell that maybe might be IGHV unmutated. That's never going to change, because that original cell, you can't go back and change that original cell. But then all of the daughter cells, let's say we call them daughter cells in that clone, are all derived from that original one. You can get additional abnormalities. So if you've got P53, then that is very likely to lead to additional abnormalities, because it's not detecting DNA damage anymore. So those things can arise. So you could start off with a 13q deletion, and then you'd look again in a few years' time, and now you've acquired an 11q deletion. So we do regularly look back and then just always compare for progression.
And should we worry about that change?
Well, I'm talking about genetics here. I'm talking about P53 and IGHV. I mean, the reality is that an awful lot of the traditional significance of these deletions is now swept away by the new therapies. So an awful lot of the significance of that is less valid than it was.
Exactly as you said, in anybody, in all of us, forget CLL, in anybody, all of our cells are dividing and changing every single day, and there's genetic changes and potential genetic damage happening in all of our cells every day. Part of our immune system's job is to fight infections. Part of our immune system's job is to recognise DNA damage happening and sort it out so those cells don't become cancer cells, okay? But for some of the cells, and as you said, we're not talking about things that can be inherited or passed on, because it's not genetic changes that are happening in sperm and egg cells that then, are something that's passed on. It's genetic changes that are happening in all of our cells all the time, and our body is trying to recognise that's a problem, sort it out so that doesn't become a problem cancer cell. But if one of those changes sneaks through and does become a cancer cell, now that's a cell with a genetic change in that Gavin can detect. And similarly, CLL cells, they're going to carry on dividing just as all of the other cells are dividing, so they also have the potential to acquire new genetic changes as time goes on.